great to be here at the Arboretum. I've had great connections and collaborations over the years with our good neighbors at the Arboretum, so it's great to be here giving a talk. So I'm also going to be talking about a rare species. My species is not quite so rare as Hinkley, but it has a very interesting distribution. It's a restricted island endemic. Vicki mentioned Quercus tomentella, species I'm going to be talking about. It's found only on the California Channel Islands and 5 of the 8 California Channel Islands, as well as an island off the coast of Baja, Mexico, Guadalupe Island. So with that distribution, that makes it the rarest oak in California. It's listed as vulnerable in the U.S. and as critically imperiled in Mexico, as there is just a handful of individuals left on Guadalupe Island. So here's its distribution. It's found so just to orient ourselves, here's the greater Los Angeles area, Santa Barbara, San Diego, the Mexican borders just south of San Diego. So these islands lie off the coast of California. Santa Rosa, Santa Cruz, and Anacapa have Quercus tomentella, as does Catalina and San Clemente Islands, and also Guadalupe Island, shown in this inset here. So just to orient yourself here on this map, this is San Clemente Island here, so much farther to the south. Just a little background on the California Channel Islands. I've been working on these islands ever since graduate school on different plants and animals, so this is a system I've always been very interested in. There's a complex geological history here, but the most important point, sort of for my purposes, is that there were never connections to the mainland. So anything that got to the California Islands had to disperse over open ocean. So what we find, just to generalize on these islands, is a rather depauperate subset of species from California and some endemics that have evolved, both plants and animals, a few species that have evolved endemic forms. Those 4 northern islands are separated by fairly shallow seas, so during periods of sea level lowering, they were connected into one large island as recently as 18,000 years ago. The biggest impact on the vegetation, including Quercus tomentella on the islands, has been Europeans introducing herbivores, goats, sheep, pigs, mule deer, bison on Catalina. They took bison out there to film Western movies and just left them. Cattle, so. But through tremendous efforts of the people that are managing the islands, most of these herbivores have been removed, and the vegetation is recovering. So this shows some of the culprits of the overgrazing that occurred on the islands, and how here's Quercus tomentella, and you just see the terrible soil erosion that occurred as a result of that. But they've gotten most of these removed, and so here's a more recent picture of Santa Cruz, and you can see how well the vegetation is recovering. Quercus tomentella is thought to be a relic species. This is what you always read when you look up. It's a relic species that was once found on the mainland. I'm not exactly sure how good the evidence is for that. There's one paper by our friend C.H. Muller that reports this, that fossil forms of tomentella are found on the mainland. It co-occurs on 2 of the islands with Quercus chrysolepis, and Quercus chrysolepis is widespread on the mainland. So we heard about this from Paul this morning. Just to review, it belongs to the section Protobalanus. So here are the North American Quercus subgroups that we now understand so much. The phylogeny has been worked out so nicely. So we have the Labadi with the biennial maturation of oaks, the white oaks with the annual maturation of oaks, and then the Protobalanus, which just is a small group of intermediate called intermediate or golden oaks or golden cup oaks, and there's just 5 species. 4 of those have very restricted distributions, including Quercus tomentella. So it was unresolved as to where these fell out relative to the major sections of oaks. But here's the phylogeny published by Andrew and his colleagues based on the RADSEQ data. So here's the section Quercus, and Chrysolepis falls out as more close related to the white oaks than to the red oaks. So just looking at this distribution, I've worked more on mainland oaks, and so what would be the expectations in terms of landscape genetics, or maybe we have to say seascape genetics for a species that has this kind of a distribution? We know that pollen moves long distances. For mainland species with wide distributions, we typically find very little genetic structure over most of their ranges. But this is a very different situation. Is pollen moving and connecting these populations? These islands are spread out, and some of them are closer to the mainland than others. So I was interested in what we would find in terms of the genetic structure of this species, and was it, really a cohesive system. So the research questions that I'll touch upon: is there evidence of reduced genetic variation for this rare island endemic? Are populations on the different islands connected by gene flow, or does each island have a genetically distinct population or something else? And finally, is there genetic structure within an island? So we collected 378 samples from all six islands where the species occurs, and we scored these for 8 or 9 microsatellite loci, and then we analyzed these data for clonal reproduction, like Janet talked about, genetic diversity, and then genetic structure both within islands and between islands. So here's just a little diagram of showing, we collect the leaves, we take them back, dry them, take them back to the lab, extract the DNA, and then PCR amplify the microsatellite loci and score them on an automated sequencer. So this is sort of what the raw data looks like, but we've heard a little bit about that approach already today. One of the first things when I went out to sample, I did some of the sampling myself, or with researchers from Catalina Island. So when we went to some sites on Catalina, we would see trees growing in these circular clumps, and we suspected that they were clones. And so we were interested in the extent of clonal reproduction in this species. So don't worry about not being able to see these numbers, but so I took leaves from 5 of these stems, genotyped them at our microsatellite loci, and take my word for it, these are all the same. So these are genetically identical across these 8 loci, and the probability of that happening by chance, if they were unrelated, is less than 1 in 10 million. So we're pretty sure they're clones. But what maybe was more interesting was that they collected some leaves for me at another site, this on Catalina, called Lone Tree Grove. They had no idea that there was clonal growth. The trees weren't growing in clumps. They sampled what they thought were 14 different individuals, but it turned out in this case there were only 2 genotypes. One represented by 9 of the stems that they sampled, and another by 5 stems. So this led us to the concerns like Janet raised for Hinklei. Is there just really extensive clonal growth in this species? Because we came upon it here at one of the first sites we sampled. Here's the overall data on that. So this shows the islands we sampled, the number of individuals we sampled on each island, and the number of genotypes that we found, the number of unique genotypes. So this is like the genet to rammet ratio shown here by this PD value. So there is clonality, but it's not ubiquitous. So we found it on Catalina. It was fairly extensive on Santa Rosa, but by and large, most of the individuals we sampled were genetically unique. You might think this is a very low sample size for Anacapa Island, but in fact, that's not That's every individual that's known on Anacapa. So it's technically not a sample. So how extensive is clonality? Well, a few stands did show this extensive clonal growth, but most recruitment was through sexual reproduction in acorns. Question about, so this is this rare island endemic. It might have been founded by just a few individuals, or there could be extensive genetic drift. Is there evidence for reduced genetic variability for this species? Here's just some overall data. These markers have been used by our lab and other labs, so we have a lot of information about variability for these loci in other oak species. And in terms of the number of alleles that occur, here's the average number per island, another measure of genetic variability, expected heterozygosity. These values are all fairly typical for oaks, even abundant, widespread mainland species of oaks. So Quercus tomentella, the good news here is that Quercus tomentella has levels of microsatellite variability that are typical for oaks, and we see no evidence for strong founder events or effects of genetic drift. The next question was about population structure and whether populations on different islands are connected by gene flow, or does each island have genetically distinct populations. So I'm going to just show you there's a lot of ways you can analyze the data to look at this. I'm just going to talk about two approaches that we used. One is a more traditional population genetic approach where I say, I think these islands are populations. I'm going to calculate a genetic distance between islands using any kind of measure of genetic distance. There are many available. The other kind of analysis is calling a clustering analysis. This is a Bayesian method. And here, the approach here is that I don't tell it what I think are populations. I just throw my genotypes into the analysis, and the analysis finds genetic clusters in the data set. So there's sort of two questions there. One is, how many genetic clusters occur among this set of genotypes? And then the second question is, and where is each individual tree assigned? What genetic cluster is it assigned to? And we did this two ways. One where it was just, the only information was the genetic information. And then another approach is where I give it both the genetic information and the location of each samples. So this is the results, or some results from the more traditional approach of using genetic distances. So here's a matrix of the islands, and above the diagonal I have Wright's FST, a common measure of genetic differentiation, and below the diagonal I've put another measure, Ney's genetic distance, also commonly used. All these values are significant. So there is significant differentiation between, but something that's a little, I see I only have 5 minutes, keep moving, that they don't make a lot of sense geographically. For example, here's Santa Rosa versus 3 other islands. Well, and these values are all about the same. But Santa Cruz is sitting right next to Santa Rosa, and its genetic differentiation is actually a little less than between Catalina and Santa Rosa. And then San Clemente is in the same ballpark, but that's one of the more distant islands. So geographically, that didn't make a lot of sense. Moving on to this analysis where we just basically say how many genetic clusters are there given these genotypes. I don't want to really get into what I'm plotting here. Those of you who do this kind of analysis know we got a strong indication that there were 3 genetic peaks in the data set. So what were those clumps, genetic clusters? Well, this is one of these diagrams where each line indicates one individual and its proportion of assignment into one of these 3 genetic clusters. So kind of what we see here is that Santa Rosa is mostly in this blue, what I'll call northern genetic cluster. Guadalupe and San Clemente are mostly in this orange. Santa Cruz, Anacapa, and Catalina are kind of a mix. But then we have this very distinct dark color here that are just some trees on Catalina Island from one site, but not all the trees from that site. So that's, I don't know, I have some suspicions about that. But then the other analysis where we actually gave it both genetic information and spatial, these were georeferenced samples, it found more genetic clusters. It found 14 genetic clusters for Quercus tomentella. So in this case, each island was genetically unique. None of the clusters were shared between islands. San Clemente had one genetic cluster, or was represented by one, Guadalupe by one, Anacapa by one, but then multiple clusters were found on the other islands. So are islands connected by gene flow, or does each island have a genetically distinct populations? Well, the genetic distances between islands were significant, but not closely correlated with geographic distance, but there were genetic clusters in the data set, and at least the last analysis, the gene line analysis, suggested that each island harbors a unique genetic cluster. And then this thing that I didn't expect to find was that here's Santa Rosa Island with 3 clusters, Santa Cruz Island with 2, if you can make them out in blue and green. And then Catalina, it'll be hard for you to see, but there's 6 different genetic clusters on Catalina Island, some very close to each other. And this is something typically you don't see on the mainland at all. I wasn't expecting to see genetic structure on this small of a spatial scale. And so this level of genetic differentiation over such a small area is rarely seen in wind-pollinated trees, including oaks. So that was a surprise. So I'm just going to end here with some complete speculation about what may be causing this genetic structure on this small spatial scale. I mean, these 3 islands that had multiple genetic clusters were the largest islands with elevational gradients and more topography, so it could have something to do with topographical barriers to gene flow and pollen movement, or there could be microhabitats that these were locally adapted to, but I don't think so. I don't think that's going to happen on that spatial scale. I speculate that it more has to do with something that these are island populations and there's a legacy perhaps of separate founding events, multiple colonizations. We also know that the Native Americans, the Chumash Indians, lived on those islands 11,000 years ago and they were a sophisticated maritime culture. We also know that acorns were a major food source, so it's possible that they were moving around acorns and planting them. What would keep populations so close to each other, separated, what would prevent gene flow? Maybe it has something to do with flowering phenology. That's the only thing that I can think of that would keep conspecific oaks that are nearby each other from some mating. So that's something I could test. Go back and look and see if there's differences in flowering time. So quickly to conclude, the landscape genetics shows that Quercus tomentella has high genetic diversity and high genetic structure. Different islands have genetically distinct populations, and population genetic structure on some islands is quite striking. And then in terms of conservation, there are major threats. The herbivores and the erosion, that's being improved now. Probably the biggest threat is invasive plant species that are ubiquitous on the islands. Guadalupe should be a focus for conservation. There's only 30 or 40 trees left, but there is sufficient genetic variability for continued recovery, and each island harbors unique genetic variation, so all islands merit protection going forward. And I'll close there with just acknowledgments of many people that helped collect the samples and the organizations that supported the sampling.